Hello and welcome to chapter 13. This is kind of a fun chapter. We're going to talk about sex-linked inheritance, so sex-linked traits. We're going to talk about different types of human diseases, things like that, and information in general. So let's get started. So first, after this chapter, you should be able to determine the relationships between sex chromosomes and sex determination. Second, understand the chromosomal basis of sex linkage, and that would be separate to see if they found more frequently in males versus females or vice versa. Third, we're going to talk more about recombination. We talked briefly about crossing over and meiosis. We're going to get into that in more depth. And then lastly, you should know consequences of non-misstructured recurrence. And we also touched briefly on that. So first, let's talk about sex chromosomes. So as we've talked about before, we have a total of 46 chromosomes, and those are in pairs. We have 23 pairs. Now, the 23rd pair is kind of a unique set of chromosomes. These are called the sex chromosomes. Okay? Now, the rest of the chromosomes are called autosomes. They don't have to do with sex. We talked about those chromosomes, right? You have two copies. They are called homologous chromosomes. They have all the same genes on them. Now, the sex chromosomes are a little bit different. As you can see by this picture, right? They are very different in shape, in length, and they do not have all the same genes on both sets, right? So the Y chromosome, you can see it's higher condensed. We'll talk about some genes that are missing on the Y chromosome as well as the Y chromosome, the X7, right? So kind of interestingly, the default for humans brain development is female, right? There is a gene on the Y chromosome that determines maleness, right? Because all of our traits are based on genes, right? And so there's a genetic basis for male or female. And so the gene that determines maleness is found on the Y chromosome. It's called SRY, which I like to joke and short for. Sorry, you're gonna be a male. Um, again, this gene is just found on the Y chromosome. It's not found on the X. So females are XX, right? Males are XY. So in the females, they just don't even have that SRY gene. Right? Again, the default development is female, unless the Y chromosome is present. So here we have a cartoon showing a female with two X chromosomes, and a male with one X chromosome and one Y chromosome, and the Y chromosome is so if you're a female, you don't have that gene at all, and again, the default is female. So this gene, the SRY gene, is responsible for masculinization, so genitalia, secondary organ formation, all the male. I just want to take a minute here while we're talking about sex discrimination and mention that there are some exceptions to this. So first of all, you can have a situation in which the SRY gene somehow gets onto the X chromosome. This would be called translocation, where a piece of one chromosome gets put onto another chromosome when it doesn't belong. And so in this case, you would have a person who has two X chromosomes. However, this SRY gene is not present. Somehow got put on the X chromosome. So since that gene is present, that individual will develop with male appearance. Another example I want to talk about is just number 3. These are mutations in the SRY gene. So if there are problems with that gene, for example, expressed, if it's mutated or missing, even though that individual is XY, they will develop female appearance because they don't have that SRY gene that we said is responsible for male development. The third type of exception you might see is androgen insensitivity syndrome. So in this case, There's nothing wrong necessarily with chromosomes. However, the body has other mutations in genes that are responsible for responding to hormones. And so androgen is the hormone that's responsible for a lot of immunity. And so in this case, if you have a signal, it has to bind to a receptor, right? And in this individual that has the mutation for that receptor, they cannot accept, the cells cannot accept that androgen hormone. So even though those cells being told have male features, they can't hear it. They don't have the receptor. And so in this case, the individual is X but will develop female features because later on down the road, that when these androgen hormones are produced, they're not affected. Also, sex discrimination is about the same across all species. So in birds, for example, we have ZW is female and ZZ is male. In honeybees, it's just whether or not their cells are we're focusing on humans, but make note, right? If you're really into birds, grasshoppers, things are a little bit different. Another exception we have has to do with mitochondria and chloroplasts. You may recall when you're learning about cell birds that mitochondria and chloroplasts actually contain their own DNA. They have their own genes. So these genes are actually responsible for keeping up the function of the mitochondria or the chloroplast. So a lot of the genes in the mitochondria are right, genes that code for parts of the mitochondria. And when you we need to think about where does the DNA come from in terms of inheritance. So when the egg and a sperm cell are made, they are like any other cells. So they have mitochondria, right? They have some organelles. The egg cell. The egg cell is very large compared to the sperm cell. And so in the cytoplasm, there's going to be a lot of mitochondria and mitochondrial DNA. Now, comparatively, when you look at sperm, these cells are much smaller. There's a lot less space for mitochondria. And so it's more likely that offspring, the zygote, is going to have mitochondrial DNA from the mother, right? This is called maternal inheritance. Great. Let's talk about the chromosomal basis for sex linkage. There's some traits that you may notice tend to be seen more often in males or females, such as red-green blindness. So if you have red-green blindness, you're probably male. And in this case, on the left up top, this circle of red and green dots shows the letter W. And in the lower right-hand circle of red dots, there's the number 2. So red-green blindness, that is actually more frequently seen in males. And on the right here, we have some pictures of my adorable cat, Annie. And she is called a calico cat, right? She's got this mixture of orange and black fur. And this calico feature is more commonly seen in female cats than males. So again, there's something going on in which these traits or these genes correlate to whether or not you have X or Y chromosomes. Again, here we've got our 23 chromosomes pointing out that the 23rd pair are the sex chromosomes, and that if you have XY, you're male, XX is female. So the first evidence of sex-linked genes was found by Pete Morgan, and so he was working with fruit fly, very common species used in genetics, and he found a mutant fly, a male fly with white eyes. Red eyes is normally seen in flies. And he crossed the mutant male with a normal red-eyed female, and he did that to determine whether it was a dominant or recessive trait. Right? He found that all of the offspring were red-eyed, indicate that this allele for red eye color is the dominant allele, and the recessive allele is the red eye color. So what I'd like to do now is draw out some Punnett squares with you, talking about these X and Y chromosomes to really get a good understanding of this, because I looked at the figure in the book and I was quite confused even after reading it several times, and so I thought you guys might be confused as well. Let's work on this sex linkage problem in flies. Let's work it out on a piece of paper. So first of all, a few reviews, right? 2 X chromosomes, right? That is going to be female. This is the symbol for female, kind of looks like a cross. And if you have an X and a Y chromosome, and I drew a line on here just to demonstrate that. Right? And I'm just going to draw the Y chromosome as shorter. Right? XY is male. This is the symbol for male, kind of looks like a shield, a spear. Right? So first thing, let's talk about the percentage of likelihood of having male or female offspring. Okay. So if we have a female and male mate, right, she is contributing, her 2 alleles are going to be, or her 2 chromosomes are the X chromosomes, and the male is contributing an X and a Y chromosome. So when we fill out that Punnett square, what we see is that there is a 50% chance of female, 50% chance of male. Right? So now with this slide, we talked about how we found that there were males that had white eyes, and he crossed that male with a female that had red eyes. Right? And this is just like what Mendel does in his system, right? To determine which allele is the dominant allele, you cross 2 strains, differing strains, and you look to see what are the offspring. And so in this case, they found that all of the offspring were red, right? They all had red eyes. So what this tells us is that red is the dominant allele and that white is the recessive allele. So I'm going to draw R, capital R as red eyes and little r as white eyes. Right? And so we know that you have these different combinations that you can get. Right? Red eyes, and 1 opportunity for white eyes. Next, what he did was he took this F1 generation and he incrossed them, right? Just like in other studies that we've seen with Mendel. Right? So what you're going to see is what we're used to seeing, right? A 3-to-1 ratio, right? 75% of these are going to have red eyes, and 1/4 of them are going to have white eyes. Now, something that makes sense, right? And that is what this scientist found as well with his flies. However, he found something really interesting. In the flies that had white eyes, they happen to all be males, right? You would expect to see that of that 25% that had white eyes, that half of them would be female and half of them would be male. And in fact, that's not what he found. He found that a greater percentage of these white-eyed flies were male. And so that's telling us that this gene has some correlation with sex, right? And so what turns out to be the case is that this gene for eye color is found on the X chromosome, right? And it is just not even present on the Y chromosome, right? So let's see what happens. What are the possible combinations that we can get? So in females, we have X, X, and they could have both dominant alleles and have red eyes. they could have one dominant and one recessive allele that have red eyes. And the third combination possible, both recessive alleles and have white eyes, right? So there's three possible genotypes that they can have. And the majority of the genotypes result in red eyes. Now let's look at the case when you have only one X chromosome, as is found in males, right? So here, there's only one allele, right? It can be dominant or recessive. So in this case, this male has red eyes. The other possibility is that on this X chromosome, they could have a recessive allele and have white eyes. And that's it. There's no third option, right? And that is because it can't even be heterozygous. There's only one chromosome that has the allele on it. And so now there's a much greater chance that the males are going to be white than red. That chance is much higher in the males than in the females. So anytime you have a gene that is found on the sex chromosomes, either the X or the Y chromosome, you're going to find that the distribution of those alleles between males and females is kind of wonky. It's not going to be 50-50. So let's summarize. When Morgan did a test cross he did an additional experiment where he wanted to make sure that white-eyed females could actually survive. What if the only reason we weren't seeing very many white-eyed females was that there was also some type of trait that killed all of them? And so he did do that cross, and he found that it is possible to get white-eyed females. I'm not going to do that cross with you, but you can find it in the book. They show the Punnett square for that. And again, what we're finding here is that if you're a male, XY, you can't have two alleles. You only have one allele for any gene that resides on the X chromosome because you only have one X chromosome. So this is called sex linkage, right? A gene or a trait is connected to sex. And so one example we've talked about already is red-green color blindness. Another example is hemophilia. Now, hemophilia was found to be really predominant in the European royal families, partially because there was a lot of inbreeding. But this is also an X-linked allele for this disease, and so a lot of males were more likely to have it. And in this disease, the mutant allele affects blood clot formation, and so they get very large bruises and little cuts or injuries that normally we could heal from. It could kill these people with this disease. The next thing I want to talk about is called dosage compensation. So you're thinking, okay, you've got some genes that are only on X chromosomes and some that are only on Y chromosomes. Does that mean that if you're a male that you only have like half of the genes level of gene like being expressed, and if you're a female you have twice as much because you have two X chromosomes? Now the body has a way to account for this, so no, we do not have double the expression of genes on our X chromosomes as females. And so what happens in female cells is that one of those chromosomes gets shut down. Basically, it's kind of tightly packaged up and inactivated. This is called a Barr body. So even though a cell is XX and it's female, the genes are only being expressed from one of those chromosomes. And which chromosome it is of those two X chromosomes that gets shut down is it's basically random, right? Let's look at an example of this in our cow cat. Right? So we're going to be looking at the cells that are responsible for whether fur is orange or black. Right? So there's an allele that will cause the cells to produce orange pigment and a different allele that will cause cells to produce black pigment. And again, this is on the X chromosome. And so basically, as I said, the two X chromosomes don't both express their genes. One of them gets shut down, and whichever chromosome gets shut down is random. So in this case, these cells, or this cell is heterozygous, so it has one allele for orange and one for black. And in this case on the left, let me get my pointer out, on the left, the one that randomly was chosen to be shut down into this bar body was the chromosome with the black allele. And so only the chromosome, only the gene allele, only the orange gene allele is going to be expressed in that cell. So it's going to produce orange pigment. Oppositely, because this is random, the chromosome with the orange allele could get shut down, and so only the black allele gene is going to be expressed. And so depending on which one gets shut down, you're going to see that some cells are going to produce black pigment and some are going to produce orange pigment. And again, because this is on an X chromosome, this is more often going to be, in this case, it's going to happen more frequently in females. Now, another thing, just kind of developmentally to talk about, is you think, okay, why doesn't this cat just have like a mixture of orange and brown, kind of like why doesn't it just look orangey brown, right? Why is it that it's patches? And so the reason that this is happening is that these cells in which this bar body is forming, those are kind of like the parent cells, and all of their offspring are going to have that same pigment. And so you're going to see, that's why you're going to see these kind of these patches. You also may say, what about the white fur, right? This is the example we talked about in chapter 12 of epistasis. There's one gene that is affecting another gene. So this cat's kind of complicated. She doesn't look like it, but there's one gene that determines whether or not you have orange or black fur. And then there's another gene that just determines whether or not there is pigment, right? And so if you get the allele that says this cell's not going to make pigment, it doesn't matter whether or not it got the allele for orange or black pigment because it's not making it at all, right? So next, let's talk about recombination, all right? So we're shifting from sex linkage and sex determination to something else, talking about chromosomes. An interesting thing is this idea of crossing over, right? And we talked about that when we learned about meiosis. So this recombination, the advantage that it provides, that you should already know, is that it promotes genetic diversity. And so I want to provide an example of this. All right. So first of all, before we get started, when does crossing over occur? Right? When does that happen? So first of all, it happens during meiosis, right? During the production of gametes. Second of all, when during meiosis does recombination occur? Right? I'm going to let you think about that one and look that one up. All right. So let's look at an example of the impact of meiotic recombination or crossing over. So during meiosis, here we've got a germ cell that has two homologous chromosomes, and I've drawn here two genes on each chromosome. Right? The G gene is responsible for body color, and the N gene is responsible for wing shape. Right? You can have gray or black body color, or you can have normal wings or vestigial wings. So in this case, both of these genes are on the same chromosome, right? So funny things are going to happen when these genes are on the same chromosome. And so let's look at what happens. And during meiosis, the first thing that happens is what? The DNA gets replicated. All right, so these two homologous chromosomes each get copied. What are their copies called? Sister chromatids. All right. Then the cell undergoes meiosis 1. These two homologous chromosomes get split apart. And then during meiosis 2, again, we skip DNA replication and go straight to the dividing up. And we have those sister chromatids finally get pulled apart in meiosis II. So here we have 4 gametes that are haploid, right? They each have one set of one of the halves of the chromosomes. And you can see the different combinations that these gametes can provide. So first of all, this top gamete can provide, it's going to contribute gray body and normal wings, right? The other gamete is going to produce black body and vestigial wings, right? And the same thing on the bottom half here. So there's basically 2 options that are being passed down. You could have either gray body and normal wings, or a black body and vestigial wings, right? So this is if you don't have recombination or crossing over. And you may say, wait a minute, we know that there are other options, right? What about gray body and vestigial wings, or black body and normal wings, right? But how can this, we know these combinations can happen, but these genes are stuck on the same chromosome. How can we mix it up, right? That is when recombination is important. So now let's look at this again with recombination. We have our germ cell, and we've got, again, the same genes. At the beginning of meiosis, first we have DNA replication. And then during meiosis I, we have crossing over. Right? So what's going to happen is a chunk of one chromosome is going to get broken off and traded with the other homologous chromosome, right? Or sorry, with the, and so with the homologous chromosome. So pay attention really carefully here to see which ones are crossing over, right? It's not the sister chromatids in which crossing over is happening. It is the homologous chromosomes, right? Now, we can see that just 1/2 of this is getting switched over. And what that is going to do is now that's allowing these new combinations, right? So now you can have gametes that are going to contribute black body and normal wings and gray body and vestigial wings, right? So again, because of meiotic recombination, we can have additional variety of genes in the gametes that are passed down, such as gray body and vestigial wings or black body and normal wings. Okay. Here is just another example of representation from your book of crossing over. Again, this is happening between homologs, not the sister chromatids. And in this, we have a nice example of kind of the physical overlap of the two homologous chromosomes. Right? They remember when we talked about during meiosis I, the 2 pairs kind of lay on top of each other, and that allows them to be really close together so that parts of them can be switched out. All right. So take some time to look at this figure as well so you really understand what the implications of crossing over are. Lastly, let's talk about non-disjunction. So essentially, non-disjunction is when a cell has too many or not enough chromosomes. And this mistake happens during meiosis. So in this example, we have a germ cell, this diploid, and we're looking at 2 chromosomes that have been duplicated already. And these 2 chromosomes during meiosis I are going to be pulled to opposite sides, right? The homologous chromosomes get split. And you can see here that one of them is going the wrong way. So if the mitotic spindle does not attach properly to the chromosomes, or if there's something wrong with it, you can have one of those homologous chromosomes get sent to the wrong side of the cell. And so then what happens is after meiosis II, you get gametes that either have one extra chromosome or are missing one chromosome. So in this case, these should have 2 chromosomes each, and some of them only have 1, and some of them have 3, right? So, aneuploidy, gain or loss of a chromosome. Um, Trisomy is the one you may have heard of, where you have an extra, right? Trisomy 3, monosomy 1, right? And again, most cases, the embryo does not survive this. And in this example, this karyotype, you can see trisomy 21. So the pair of chromosomes, the 21st pair, instead of being a pair, there is a third chromosome. Now, some of these chromosomes that are smaller, if they get a third copy, they may not have a large effect on the individual. They may be able to survive. And so one example is trisomy 21 or Down syndrome. So this could be a situation where there is an entire third 21st chromosome or possibly a translocation where part of chromosome 21 gets added on to another chromosome. And we know that the mother's age influences risk. I'd like just to stop a minute just to this is an interesting point. Remember when we talked about when meiosis happens in females and when it stops? So we talked about how meiosis 1 actually occurs during fetal development, and that not until puberty does meiosis 2 begin. And meiosis 2 lasts until menopause, and at menopause, meiosis stops completely and no more gametes are formed, no more eggs are formed. And so it's likely that as the system is shutting down, that things are more likely to go wrong, and that is why trisomy 21 can happen more often if the mother is older. So we can also have non-disjunction of the sex chromosomes. So earlier, chromosome 21, that's an autosome, right? Doesn't have anything to do with determination of sex. And interestingly, most individuals that have non-disjunction of X chromosome are able to survive, and some of them may be fertile as well. And so here we have a lot of different combinations of non-disjunction that can occur. So trisomy, right, where you have 3 chromosomes, or monosomy, where you only have 1, and so the O just means none, right? And so you may have heard of Klinefelter syndrome, or Turner syndrome, or Jacob syndrome. Here I'd like to show an example of kind of how this can happen. So in this case, we have female gametes underwent non-disjunction. And so what we end up have happening is that you can have 2 alleles, 2 copies, 2 chromosomes being sent in one gamete and resulting in trisomy, right? We've got 3 X chromosomes. Klinefelter syndrome, that is when you have 2 X and 1 Y. There's the possibility for Turner syndrome, and then also this last one where you just have the Y chromosome, that is going to be non-viable. That's it for today's lecture. It's really short, but there is going to be an activity associated with this lecture. And you're going to have to get out some paper and pencils, maybe some tape and scissors, and we're going to play around with meiotic recombination or crossing over. And so be on the lookout for a separate video that talks about what to do. And then there will be an assignment where you're going to have to kind of report on your activity that you did. Thanks.